Hello everyone, I hope you're all good. From Avery's experiment, it is clear that DNA is the genetic material, but the problem was every scientist have not agreed it. Now, the another experiment which was conducted by Hershey and Chase, popularly known as Blender's experiment. I'll explain why it is called Blender experiment a little later. It has given the unequivocal proof, that is, after this experiment, no other objections were given for the hypothesis that DNA is the genetic material responsible for transformation of R strain of bacteria into S strain. Now, this experiment was conducted by two scientists, Alfred Hershey and Martha Chase. So you have to remember the names of scientists. Alfred Hershey and Martha Chase has conducted experiment during the year 1952 and they worked with bacteriophage. You have studied from first year that bacteriophages are the virus which infect bacteria. The bacterial infecting viruses are called bacteriophages. Now, to understand this experiment, first you should know the life cycle of bacteriophage. That is, it is also called T4 cycle. Before that, briefly we should know the structure of bacteriophage also. Bacteriophage is having a very simple structure, having a protein coat within which genetic material is present, and this genetic material could be DNA or RNA. Remember this point, genetic material could be DNA or RNA, because at last I'll ask you one question or a doubt something, so that there you have to remember this point. Now, during the life cycle of bacteriophage, the bacteriophage first attaches to the bacteria. In NCERT also they have given the point in brief. Bacteriophage first attaches to the bacteria, then its genetic material will enters into bacteria. The genetic material could be DNA or RNA. It will enters into bacteria. Now, after entering into the bacterial cell will treat the genetic material of bacteriophage as its own, and it will start synthesizing the materials which are required for the synthesis of new bacteriophage. So, as many bacteriophages are synthesized within the bacterial cell will undergo lysis and releases the bacteriophages into its surrounding, and those bacteriophages will start to infect the other bacterial cells. And this is in brief about the life cycle of bacteriophage, or it is also called bacteriophage cycle. Now, let us understand the experiment. That is, main focus will be on our Hershey and Chase experiment. Hershey and Chase conducted an experiment to prove whether it is DNA or protein responsible for or which is transferred from virus into the bacteria, because the bacteriophage is having two components, DNA and protein coat and DNA, genetic material will be present. Among them, which one is entered from the from bacteriophage into the bacteria. Now, to do this experiment, they have grown some virus or bacteriophage in a medium containing a radioactive phosphorus, namely phosphorus 32. This is also very important, which radioactive element is used to label the DNA. So phosphorus is used to label the DNA because phosphorus is present only in DNA and it is absent in protein. This is very, very important point. Hence, phosphorus will be incorporated into the DNA of a bacteriophage. Now, the protein coat is not having any labeling, whereas the DNA will be having a radio labeling with phosphorus 32. So, this is also given in diagrammatic representation. So, you have to remember DNA is labeled with phosphorus 32. On the other hand, other virus or bacteriophages are grown on a medium containing the radioactive sulfur 35. You should remember here, sulfur is a component of only protein and it is absent in DNA. That's why they make these two elements for their experiment. Otherwise, they would have choose some other, because phosphorus is present only in DNA, sulfur is present only in protein and it is absent in DNA. Now, sulfur will be incorporated into the protein coat of bacteriophage. Now the protein coat is having labeling with sulfur 35, whereas DNA will not be having any radio labeling. So remember, these two are the separate experimental setup. It will be conducted separately and they will compare for the result. Now, the further experiment is conducted in 3 steps. The first step is infection. During infection, the bacterial cells are attached with a bacteriophage, or else bacteriophages are allowed to attach with the bacteria. Now, the experimental setup will be separate. You should remember this. Don't mix it up. So, in both the cases, the steps are 3 and the process will be same, but whatever the result they obtained will be different. But I will be explaining them together. Now, after attachment of bacteriophage to the bacterial cell, the genetic material will enter into the bacteria, because we have studied already the life cycle. First, genetic material will enter and many of the viral particles will be synthesized. Now, here the life cycle will not be studied. Here the process is half. So I'll discuss that later. Now, after attachment of bacteriophage to the bacterial cell, the genetic material will enter into the bacteria. Now, the life cycle will not be allowed to continue here. Now, the viral coats will be on the bacterial cell, whereas genetic material will be in bacteria. Now, these two are separated. That is, the viral coats of bacteriophage are separated from bacteria by the process called blending. Here, the mixture will be kept in a blender and hence it is called blender's experiment. Blender is nothing but simple mixer grinder as we use in our household purpose. That's why it is called blender's experiment. Now, after blending, bacteriophage protein coats are separated from that of bacteria. Now, in both the cases, bacteria is separate and bacterial coats, the bacteriophage coats are separate. But remember, both of them present together, not attached, but they are present in a mixture. So, I hope you can imagine here now. So, within the mixture, there is presence of bacterial cells also, there is presence of viral coats also. They are, even though they are separated, they are present together in a mixture or in a solution. They will be present. Now, these are subjected, the solution is subjected for the process called centrifugation. Centrifugation is a technique where the components are kept in a test tube. So, by keeping them in a test tube, it is kept in an instrument called centrifuge. Now, by applying a centrifugal force, the larger particles and smaller particles present in a solution are get separated. Now, there is a separation of two fragments. The fraction which is separated above is called supernatant and which is sedimented at the bottom is called sediment. Now, in both the cases, the same thing. The above portion is called supernatant and the bottom layer is called the sediment. And here one more very important point. In this case, in the solution, there is only two components. One is bacteria and another will be the viral coats. The sediment always contain heavy particles. Supernatant will always contain lighter particles. In this experiment, heavy particles used are bacteria and the lighter particles used are viral coats. So, they are get separated. So, now you recall the point. Sediment, what it contain? Bacterial cell. And supernatant, what it contain? Viral coats. Now, in first set of experiment, that means the viral DNA which is radio labeled. In that case, the sediment, that is bacterial cell, have showed radioactivity. That means what? The radio labeled DNA, that is with phosphorus 32, has successfully entered into the bacterial cell. That's why bacteria have showed radioactivity, which means what? DNA was the material that has passed from bacteriophage into the bacteria. Hence, DNA is the genetic material. But on the other hand, in second set of experiment, bacteria did not show any radioactivity because protein coat is labeled over there and protein has not entered into bacteria, hence bacteria did not show radioactivity. If protein is radiolabeled, then if protein is the genetic material, then bacteria would have shown radioactivity. Here protein is not the genetic material, hence bacteria have not showed any radioactivity. Therefore, from this experiment, it is unequivocally proven that there are no objections, some more other objections for this experiment. So that was proved as DNA is a genetic material. Fortunately, Hershey and Chase used a bacteriophage which is having DNA as their genetic material. Just think, what if the bacteriophages were having RNA as their genetic material they used? So everyone will be believed that RNA will be the genetic material, isn't it? So think over it and I'll come up you with another video in next